Thanks for the opportunity to give this talk. And today I'm going to talk about how to evaluate the impact of genetic variations, particularly on transcription factor binding. We talked a lot about the variations in the non-coding regions, and people say a lot they might be associated with transcription factor binding sites. Here we call the TFBS, and a lot of known literature shows that the disease-related variations are enriched in the transcription factor binding regions. And this is really my goal, to predict whether certain variations would impact the specific transcription factor binding. And the first key question for me is that if you want to build a statistical model, you need to collect enough data of the disrupted binding sites. But when you look at the literature, there are very limited regulatory SNPs with validated impact on the TF binding. Here are several literatures, usually less than several hundred, and so this is totally not enough for build a good model. And in this project, we focused on another type of data, which we call allele-specific binding, or ASP. In the human genome, we have 2 copies of DNA here, and then we would have some heterozygous sites. And if transcription factors significantly prefer to bind one of these allele, we would call it as ASP events. The ASP events is good for our purpose to predict the impact on transcription factor binding, because it provides the quantitative binding difference between these 2 alleles. And the second, these 2 alleles are in the same cell context. So when we compare the nucleotide change, they are in the same cell context. We are not biased by the cell context difference. And so to collect the ASP data, we developed a pipeline which requires the whole genome sequencing data and the ChIP-seq data. First, we would use the whole genome sequencing data to call the genotype of the given cell line. And then we use the code SNPs to build a personal genome. This personal genome would enable us to better map the reads, cotton non-reference allele onto this cell line. And then we map the ChIP-seq reads to this personalized genome, and then we would extract the binding signal at each allele. And if some, then we can compare the binding difference. Here it's similar, we call it as non-ASV. Here there are some significant difference given by the binomial test. We would call it as ASP events. And here are the data we collected from the ENCODE project. Overall, we collected 45 TF ChIP-seq experiments. And actually, only 0.4% of the ChIP-seq peaks are classified as ASP. Most of them are non-ASP. And this is the total number of ASP, about 10,000 we collected. And the next, after we have the data, we look at the potential features. The first feature is the traditional one, PWM. It reflects the binding preference of the TF. And when we compare the PWM score of the favored allele, which means higher rates of the ChIP-seq, and the unfavored allele means lower, we found that in ASP events, the favored allele always show higher PWM score. So the motif disruption for the unfavored allele kind of caused or related to the transcription factor binding disruption. When we look at the non-ASP events, we saw quite balanced, and so the PWM score difference is quite informative for the ASP events. Next, we further look at the DNSI hypersensitivity, DHS, and quite similar. We found that for the favored allele in the ASP events, it shows more openness. And for the non-ASP, overall, it's quite balanced. We further measure this correlation use allele imbalance. Basically, it's the read count of one allele over the sum of the total read coverage. We can do the correlation of TF allele imbalance versus DHS allele imbalance, and you will get a very significant p-value. And we further did the allele imbalance across all the TFs of our compiled ASP data and all the available chromatin properties, including 1 DHS and 11 histomodifications. And you will see a lot of features, mainly including the DHS and several active histomodifications are correlated with the ASP events of TFs. Particularly for the DHS, this is the most widely correlated one. And after, at this point, we have a lot of features correlated with ASP events, and then we go to a classification model to incorporate all these features. So it's quite straightforward. ASP means impacting, and non-ASP means the negative set. And then, based on the features, we developed 3 models. The sequence model only collects the features based on the PWM, including the chipped TF, including the position of the SNP, whether it's overlapped with the predicted TLBS or not. Also, we included the other enriched motif. This is a sequence model. For the DHS sequence model, we just add DH data to test what's the efficiency of the DHS. And the full model, we include more histomodifications and whether it's overlapping other TF binding regions. For each TF, we trained these 3 models using random forest. And here is the cross-validation accuracy of all the 39 TF data sets. Basically, you will see the full model have a lot of features, would outperform the DHS-seq and the sequence model. And the DHS, however, the DHS-seq model also have a very significant improvement compared with this one. It almost covers 60% of the improvement compared with this full model. So our conclusion is that adding the DHS is quite cost-effective if you want more accuracy. So here, just a summary. First, the ASP approach provides us a high throughput way to collect altered TF binding events. And the second, the ASP classification model can enable us to incorporate as much as features you want to predict the variation impact on TF binding. And the third, adding DHS data to the whole genome sequencing is quite cost-effective. Yes, that's it. I'm ready for questions. Oh, sorry. Here I'm going to thank my supervisor, Vice Wasserman, and the lab members of our lab. Because in this study, we use the Heller S3 whole genome sequencing data. So I'd like to thank Harry Anna Lex. This is the donor of this cell line, and this study was under-reviewed by this NIH Heterogenome Data Access Working Group. And I thank the funding resources, and I'm ready for the questions.
It's a very nice talk. So I'm sort of curious whether you can distinguish between which transcription factor motifs mediate the DNase hypersensitivity and which do not. And does it make sense biologically what's known about these transcription factors?
So you want to ask which one is the reason or which one is the result? TF binding.
So which motifs correlate with a loss of chromatin accessibility? So these would be the sort of pioneer transcription factors. And so I wonder whether your data can actually reveal a sort of an assay for what could be a pioneer factor in just increasing accessibility in general, and which ones are very specific bindings that just abolish sort of a very particular motif, but doesn't affect binding of other factors.
Yes. Actually, I didn't figure out a way to test the dependency between TF binding and other chromatin properties. And there are some papers talking about the disruption study has a potential to reveal the dependency, but I fairly don't know how to do it, and maybe we can discuss it. Yeah.
Actually, in Pritchard's 2012 Nature paper, where he measures chromatin accessibility and defective genotype, he showed that it's positively correlated with a large set of allele-specific.
Or David Gifford's paper?
No, Jonathan Pritchard.
Oh, Pritchard, okay.
And Degner and Pritchard.
Pritchard.
I don't know.
Yeah, other people have done so. Yeah, that's why I was curious. In
Yeah,
your case,
but that's still correlation, not the. Yeah, but not the real dependency.
No, I'm not asking for experiments. I'm just asking what you would predict based on your data. Strong correlations, yes.
I think for just we need to classify transcription factors. Some ones are at the top layer of regulatory networks. These ones might impact the DNase directly, and others would just follow the DNase pattern. Yeah, sorry.
Yes.
Thank all the speakers in the session.
Thank you.